Good, so test two. Measuring transpiration. I need to measure a volume per an area of leaf per a time period. So on this test, it is good old D, but it does depend on what test you had. But yeah, it has to be millimeters cubed is a volume, and then yeah, because we're comparing different leaf sizes. Question two is just, do you know your mean, median, mode? Again, it sounds easy, but people get these wrong in the exams. I'd have a quick check. Look at my modes. Well, it's either 12 or 18. How many 12s have I got? I've got 1, 2, 3, 4 12s. How many 18s have I got? I've got 1, 2, 3. Right, so it's either the mode is 12, so it's either B or D, so it's not C. So let's do the median next, because that's probably the next speediest ones to do. So cross off a low and then cross off the 12. So I've got 1, 2, 3, 4, 5 I've crossed off there. Let's cross off 5 high ones. 20, 19, 18, 18, 18. That means I've got 14 left and 14 left. So it must be D. Yeah, a mode of 12 and a mean of, sorry, and a B. It must be B. Yeah, 12, I was looking at the mean column, and 14, yeah, so it's B. Good. Hopefully an easy start. Unit conversion. I always like to write down what I have before I start, just to kind of make sure I know. So I've got 30 millimeters cubed per minute per, let's put per 37 centimeters squared, because I need to get it in centimeters cubed. So I need a millimeter cubed into centimeter cubed. It has to be in per hour, not per minute, and it needs to be per centimeter squared, not per 37 centimeters squared. So let's do the millimeter to centimeters cubed, millimeters cubed to centimeters cubed first. So yeah, so 1 cm cubed equals, well, that's 10 by 10 by 10. 10 times 10 is 100, so that's 1,000 millimeters cubed. So if you divide 30 by 1,000, again, I don't even trust myself. I was chucking an extra decimal place. Yeah, so that equals, yeah, so it's 0.03 centimeters cubed per minute per 37 centimeters squared. Let's get rid of the 30 centimeters squared as well. So I have to divide by 37, because 1 centimeter squared of leaf is going to transpire less than a bigger area of leaf. So now I've got my 8.11 times 10 to the minus 4. And now I'm in centimeters cubed per minute per centimeter squared. That's good. So now I've just got to get it into an hour. So in a minute it loses that much water. So it'll lose more water in an hour. How many minutes an hour? It's 60. So multiply by 60. So my final answer is, yeah, 0.0486. It needs to be in standard form, 2 sig fig. So shift the decimal place across a couple of times. So it's, yeah, 4 point, oh, 4 point, it's 2 sig fig, isn't it? So so it's 4.9 times 10 to the minus 2 is the answer that I get. I'm as likely to be wrong as a maths question as anyone, so hopefully I'm right there. I'm not using the mark scheme, I'm just having a go at these. Diploid number of the cell. It says it's a diploid cell. It is just, I've got 1, 2, 3, 4, 5, 6, 7, 8, 8. I've got 8 chromosomes. It's the number of chromosomes. Admittedly, there's 4 pairs, but you just count the number of chromosomes. This cell has got 8, and that's its diploid number, because it's a diploid cell. Drawing this, well, I just need one of each homologous pair, and look how they've given different shapes and sizes. So I just have to kind of like roughly match that. So there's one small one, there's one kind of like longer one, there's one kind of like quite big bent one, and there's more like a hockey sticky one. There, I'm done. Yeah, as long as I've got 4 chromosomes shown for 1 mark, and trying to get one of each pair. So I've taken that one, that one, that one, and that one there. Again, nice and easy. I don't have to show them in a certain orientation. Number 6, the point of meiosis that results in exchange of DNA between homologous chromosomes. Well, that's crossing over. You can argue, because it's my wording of a question, that happens during prophase 1. You don't need to know that, though. You should know crossing over. Yeah, homologous chromosomes come together. Again, I did a video on that for the second revision lesson, so why not have a watch of that one? You've got the email. Number 7. So one of you had 7 pairs of chromosomes. This one's 8, and it's always 2 to the power of the number of chromosome pairs. So if I've got 8 pairs of chromosomes, it's 2 to the power 8. And 2 to the power 8 equals 256. Yeah, so 2 to the power 7, which some of you had instead, was 128. Yeah, in a human cell, it'd be 2 to the power 23, which is 5 million and something. And then 8, which one of the following will result in a non-disjunction mutation? So one of the bases is substituted. No, that's a substitution mutation. A frame shift results in a different sequence of codons in the mRNA. No, that's due to an addition or a deletion. Cytokinesis fails to take place during the second part of meiosis. Well, that would just result in a cell with far too many chromosomes. That's not non-disjunction. Yeah, it's D. It's when spindle fiber fails to separate centromeres of a chromosome. Therefore, one of the chromosomes stays together, and then that goes to one of the poles, and therefore you've got 2 copies of that chromosome in there. So it is D for this example. We do meet mutation again in topic 8. Number 9, 2 plants were selected. Some of one was ringed, the other was unringed. They were given radioactive carbon dioxide. You can see it here in this transparent box. We can still do photosynthesis. And you might know a bit more about this now, because it's kind of similar, well, a bit similar to what Calvin did for his lollipop experiment, apart from he used algae instead. And then, figure 2 below is showing reactivity. Which of the options explains how radioactive carbon from carbon dioxide supplied reaches the buds at the end of the stems? So the bud's at the top. You can see down here it's gone to the bud, and it is. Let's have a look. Glucose made in photosynthesis is transported in the xylem. No, it's not. Sucrose is transported in the phloem. So that one's wrong. Just shimmy it across a bit. That's just a bit in the way. Carbon dioxide is transported in the phloem. Carbon dioxide is not transported in the phloem. Gases are not transported in plants. Translocation of sucrose occurred. That sounds good. Let's just read the other ones in case. Radioactive carbon dioxide dissolved in the sugary cell sap. No, because the carbon dioxide would have been used in photosynthesis to make radioactive glucose. Then the glucose would have been joined on by glycosidic bond onto fructose to form sucrose. So that's no. And radioactive carbon dioxide diffuses into pores near the bud. No, because you can see here it is isolated in this transparent box. So even if E was possible, if buds had pores for some unknown reason, which they don't, it can't be E. So it is C. Question B, hopefully did this and didn't miss it out. Mark an X on the ring plant in figure 2, the position at which the stem was ringed. Well, I need to know the ringed plant first of all. It's the right-hand one, because that's got 0 radiation here and here. So I've stopped the radioactivity moving. So it must be, well, again, and that was the leaf that was fed or given radioactive carbon dioxide. So I'd be putting it around here. But anywhere in that zone there would count. And it says mark with an X on the ring plant, so I should have done exactly like I've done there with my X. And question C, which 2 options below explain the results in the ring plant? Why has it not got radioactivity below the leaf? And I need 2 of these. Ringing removes phloem. It does. That's the definition of ringing, so A must be one of them. osmosis can't occur between xylem and phloem. That's incorrect because the xylem and the phloem are still next to each other. I just stopped part of it. I've just removed a portion of the xylem, sorry, the portion of the phloem in one region and left the xylem intact. So that's not correct. Transpiration cannot occur. No, the xylem is still intact, so that's not true. Radioactivity, that should be from, not for, I'm sorry. From 14CO2 has damaged the phloem. No, it hasn't. Sucrose from the leaf can't reach the roots. Yes, it's E. Yes, or yes, sucrose just can't get past here because the phloem's broken. That's how it moves. So hopefully you didn't get that wrong because of a form from typo. Well, fingers crossed. Bit of taxonomy. An order is a taxonomic group. First thing I write down here is I get my king prawn curry or fat greasy sausages. So I know the order, literally the order. Yeah, the order comes here. Oops, oh, pen jumped. Yeah, order's there, nice in the middle. And as I go head down here, I get more and more specific. So there's only, I'm showing 1, 2, 3, 4, 5, 6, 7, 8, 9 different species. And it says an order is a taxonomic group. All seals belong to the same order. Okay. Name one other taxonomic group to which all the seals belong. So I have to go the other way. So I could say kingdom. So they're all in the animal kingdom. They share that. So I can say kingdom. I can say phylum. They're all chordates. They all have backbones. And they're all in the same class, which I think is mammal. I'm assuming I'm right there. Phylum, actually no, the phylum's probably mammal. Anyway, you don't need to know any classification of any organism. I'm not a seal expert. Then 11 and 12 refers to the diagram. How many different genera are shown? A genera is the genus, and I would just say, right, well, I've got genera is always in italics but capitalized. So they have the same genera or same genus. And those three are the same genus as well. They're all that one, and then I've got 1, 2, 3, 4 different ones at the end. So I've got 4, 5, 6. There are 6 different genera shown. 9 different species, but only 6 different genera. And then all the seals shown in the diagram are members of the, whatever that word is. Which taxonomic group is that an example of? Well, I've got my species there, I've got my genus there, therefore the next branch out is F. So it's family. The family is that one. And then we get onto a bit of courtship. So different techniques are carried out to measure how genetically different these two things are. Which one of the following would not provide valid data? So let's do our tick crossing again. Comparing the primary structure of proteins, well, that would give me valid data because I'm comparing proteins are coded for by genes, and therefore that would give me valid data. If it's a change in a protein, there'll be a change in the primary structure of the protein, as in the amino acids sequence, then it reflects on the genes. And introns are between genes, and they pick up mutations, and therefore they are really variable between individuals. So they are really useful. Exons are useful as well, because exons will give you different amino acids and therefore different protein structures. So they're all super for comparing. Comparing which codons code for which amino acid? No. This is the universal code. Universal code. So it's the same in all life, bar some the archaea bacteria. So it must be D. Just take comparing the tertiary structure of plasma proteins. No, we do that with like immunological techniques. So it's definitely D, because that's like the opposite. It's the same thing about life. It shows that we're all like related because we all have the same like codon map. AUG codon always codes for methionine. in all of life, again, apart from these archaea bacteria that branched off really early. Mole crickets, who knew, are insects that live underground. The courtship song, it's a bit of maths, but again, relatively simple. It's like, do I use my ruler or not? Well, my keys here, so 2 of those blocks is 0.5 of a second. It's species F, so I'm on this diagram here. How long does one song take? So there's 2 units there, so that's 0.5 a second, that's 1 second, that's 1.5 seconds, that's 2 seconds, 2 seconds. Again, it seems too easy for A level, but the number of people who, I've not seen the examiner's report for this, but I bet many people pick G, not F. I don't know how they go wrong, but yeah, just misinterpreting a scale. I guess it's a scale bar type thing again. And then the rate, it's a bit like doing heart rate of this or breathing rate. All I do is do 60 seconds divided by the song, which is 2, and therefore that's 30. I get 30 songs per minute in species F. There's no like gap in between. It just goes song, 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 song. Which option below is not a reason why species have courtship rituals? Yeah, not a reason. Again, you have to, well, pair bonding. No, they do it for pair bonding. To ensure they are the same genus. No, it's to make sure they are the same species. Well, let's read them through in case there's a better wrong answer. To ensure breeding takes place between fertile adults. Yeah, because sometimes you don't want to mate with a juvenile who's not fertile yet, who just looks like an adult. In the in this example, to ensure offspring are fertile. Yeah, because that's when members of the same species produce fertile offspring, and to synchronize release of gametes. Yeah, if you're aquatic or something, you need to make sure that you're releasing gametes at the same time. So yeah, it's definitely B. So that word genus, it should be species, and then there'd be none wrong if it said species. So question D. Yeah, I've typed this one badly, but yeah, we've moved some crickets from a grassland onto sand when we're measuring the colour, and it's undergoing directional selection. So that means that the colour is going to change. So it's definitely not D, E, and F, is it? Because that's no change in colour. So it's not any of them. Yeah, you'd expect the mean colour in the population to become increasingly not green or like yellowy or something. So I've done that one, and then I've got a choice of standard deviation, decreasing, increasing, or no change. Well, it is C. Yeah, you just get a shift in the shape. It's going to go from a color value where it's kind of like green over here and yellow over here. So the original population was there, showing a normal distribution curve, and then it's just going to shimmy over here over time. Therefore, yeah, like there's no reason for the actual shape of the curve to change. The normal distribution curve, it's just going to shift. If standard deviation decreased, well, that's an example of stabilizing selection. If it increases, that's one we haven't done yet. That's disruptive selection. That's part of a future topic. So yeah, so it must be C. Yeah, you get a change in the color value, and the standard deviation would stay the same. Apologies for the wording being a bit bad, but you could definitely work it out anyway. And then we've got this snowshoe hare. So somebody had a mouse, somebody had a snowshoe hare. It's the same question though, because it's all about fur colour changing. And we allow that if you don't know if it's that this is an animal that's fed upon by a carnivore. You can word it in different ways, but I'm going to go through the main ways in terms of as in the correct answer, but yeah, different interpretations are allowed. But crucially, for the first marking point, you need to talk about that there's been a mutation. So there's a mutation causes a new white fur allele in this example. Yeah, white fur causing allele. You need to talk about mutation in an individual causing the white fur. That's our first marking point. Next one, you say about how being white aids survival. Yeah, and really, if it's white fur in the winter, you don't need to be too much of it. I know it's an Arctic habitat, right? Well, it's probably a camouflage thing. Yeah, it's snowy, and therefore, yeah. So yeah, those with white fur. So these few mutants, I can't spell with white fur, have better camouflage. Don't forget the U in camouflage, therefore, are less predated. So if you put there, can gain food more easily or lose less heat, you'd still be okay, but it is a camouflage thing. Yeah. Or I spelled that wrong now as well. Less predated. Less predated. Oh, come on. Predate. Or even better is they have less predation pressure. Yeah, it's very much the same as GCSE is this. It's just wording. You need to be careful. So these then survive to breed. We need the word breed or breeding. And pass on their allele. Pass on their white fur allele. You have to say allele to their offspring or young. But yeah, you can't just say, you can't miss out any of those words. Breed, allele, and offspring need to be clear, or synonyms can be used, but breed, allele, and offspring are better. If you just say, yeah, these survive and breed and pass on their white fur to their offspring, too vague, miss off allele, it's genetic. These survive to breed and pass on their white fur allele. Yeah, who to? You need to say to their offspring, it's not just enough. Or these survive and pass on their white fur allele to their offspring. How did they do it? They do it by breeding. And it sounds obvious, but it's the number of people who miss it out. And then it's just how they evolve. So it's just saying what evolution is, and therefore, yeah, the frequency of the white furred allele increases in the population. Yeah, so you have to say that the allele, this becomes more common. Yeah, again, normally people get this wrong because they get mutation. They make it sound as though it's mutated because of the advantage. It's just a random mutation. People get this wrong because they will just say it provides better camouflage, and they don't want to say why it's good to be camouflaged. As I painfully went through, breed, allele, offspring need to be clear or similar words, and then evolution is a change in frequency of alleles of genes over time, and in this case, because it's about a specific allele that causes white fur, this population is going to be increasingly show this white fur trait, and therefore the white fur allele increases in the population. Super. That's paper 2 done. Hope you found something useful.